Welcome to the Jack Westin MCAT podcast with your host, Phil Hawkins.
And Azaii Calderon-Muñiz.
All right, we're back again, and we're going to shift gears. I know we've been talking a lot about psychology for the last several episodes in the podcast, but I think it's time for us to get back to the basics of biology. And when I say basics, I mean really the basics, the central dogma of how it is that we exist as humans, the central dogma of biology, which is how we can take DNA, turn that into RNA, and turn that into proteins. Overall, this is one of the highest yield topics. It shows up in lots of different ways. There's no way that we're going to be able to talk about this all in one episode. What's going on with transcription, translation, what's going on with bacteria, how we control and modify these things. These are all different topics. And we're going to spend a couple of episodes really diving into this because we know that this is a fairly high yield topic. And it's something that a lot of students feel pretty confident about. But then when they take a test, they find out this is being tested in weird ways and exceptions to the rule are being tested. And so it requires you to really understand very well.
Yeah, and it's also really easy to get the terms mixed up. And I know that in the past when we've been at this, both of us, for quite some time. And in the past, when I was tutoring or doing some science sessions elsewhere, the 5', 3', the numbers would get students all sorts of mixed up. And I remember when I was studying, that was also the case. And so as we're talking through what happens in transcription, translation, I don't think we would talk about replication, but we can think about that, or rather students, we recommend that you guys think about that. And also we're going to try to show some images for the folks that watch us on YouTube during some of these, but we really recommend that you take a look at some images, especially if you're a visual person. I think that would be a really great supplement to these upcoming podcast episodes. So we're going to talk about things like control, like you mentioned in future podcast episodes. Same with eukaryotes, prokaryotes. But maybe we should just dive into the different stages, particularly in eukaryotes. So this episode's going to focus on eukaryotes.
Yeah. And by eukaryotes, we mean you, because you are a eukaryote. Yeah. So eukaryotes are basically just not bacteria. And so things with organelles like we have. Now note that I just want to be kind of careful here. A lot of times people think prokaryotes means single cell things. There are single cell eukaryotes. And that's one of the ways that the MCAT can be really tricky is they'll test you about a single cell organism that might be a parasite or something. And it is a single cell, but it's still a eukaryote. So this isn't just humans and then pathogens or something else. There are pathogens that are eukaryotes. And so to start off with, we want to talk about transcription, right? I guess kind of in a way, taking a step back for just a moment. I like to think about transcription and translation, this whole process. Because they're designed to help us build things in our body that we need, right? And so in our cell, we have this giant library with all these books with the blueprints for all of these different things. And every single cell in my body, they all have the same library. That doesn't mean that every cell is making every single thing in the library, right? The library might have designs for chairs and cars and airplanes and this cell just needs chairs. And because different cells in my body need different things. The cells that build bone are different than the cells that help my heart contract, which are different than the cells that secrete insulin. And so we have this giant library, and imagine that in this world where I've got one copy of these blueprints, right? This book that has the blueprints for building this thing. The people in the library aren't going to want you to take that blueprint out and take it to the factory, because what if something happens to it while it's out there? And so what happens is, so your body won't take DNA to your factories, which is ribosomes, so the things that are building stuff. Your DNA stays in your nucleus. It does not leave. But if we want to get that message, the blueprints to the ribosomes, we need to make copies of it. And to make copies of those blueprints, that's transcription. To transcribe literally means to make a written copy of something that's over there. So like I would look at the book, see the blueprint, make a copy on a piece of paper, and then that piece of paper I can take to the factory and be like, okay, this is what we want to do. Now by piece of paper here, the analogy is RNA. So we have DNA stays in the nucleus. RNA is like a temporary copy of our DNA that we can take to the ribosome to build. And so transcription is this process of making that temporary copy.
Yeah. And there are so many different, or not so many different, but there are a couple of different stages in eukaryotes, how this transcription ultimately happens. And so thinking about the different stages, there are 3 main stages. And you'll actually hear these terms with translation as well. And so that's another way that it can be, we have to be careful when we're answering questions on the MCAT. And so the first stage that we're going to be talking about, and we're going to go through these, so I'll just mention them, but we're going to be going through this in depth for each of the stages. The first one is going to be the initiation stage. And so that one, the name is pretty straightforward. The names of all of these, I think, are pretty straightforward. And so that's going to be where we start actually making the messenger RNA or the mRNA, which is the specific type of RNA that is that paper copy, like you were saying, because we have lots of different types of RNAs. And we're going to talk about some of these other types of RNAs, both in the following episode and maybe a little bit more down the line when we talk about the control of different, when we're actually making these different proteins. And after that, we have elongation. And so this is where, again, we have to be really careful comparing it to translation. And then we have that termination phase and different modifications that we can make. And this is another place where it can be tricky because we learn some terms that can actually trip us up when we're answering questions on the MCAT. And so there are some proteins that help us out when we're starting off the transcription process.
Yeah. And I kind of like, I use this analogy and I kind of am like, okay, we need to be like super clear on all of this stuff. I know you just mentioned translation, transcription, we're kind of talking about now. So transcription is making that messenger RNA. Translation is translating that copy of the blueprint into the actual protein. And so when you mentioned, we'll talk about translation later, that's what we're talking about, is turning this paper copy or this copy of our DNA into an actual protein. But in this like initiation step, if we're going to be making a copy of this, we need to unzip our DNA, because the way that DNA works is we have like one strand and then we have another strand that matches up with that. And so whenever we make a copy of that, we need to pull them apart and take one of the strands and make a copy off of one of those strands. And so in order to deal with this, we're going to need to unzip the DNA. And so there's an enzyme helicase, which is able to unzip the DNA and kind of pull them apart. I remember my biology teacher in undergrad taught us all this pickup line for biologists, and I'm going to share it, even though I don't know if I should. But the idea being, you walk up to someone and say, like, if you're an enzyme, then I'm helicase, so I can unzip your genes, which is, I kind of, part of me cringes at that now. But as a result, I always remember helicase. Helicase is a very interesting enzyme. It actually like spins incredibly fast, and it like kind of like rips apart. I don't want to say rip because it implies damage, but it like pulls apart those strands of DNA, and helicase is turning almost like a jet engine as it's like going through this and pulling apart the two strands. But there's also topoisomerase. This is making me feel old, because the only way I've been able to understand topoisomerase, and like when I was younger, is like talking about corded phones that people would have in their houses. And so like whenever you unzip the DNA, it can create something known as supercoils. And so if you've ever had like a rope, right, or a cord for a phone, or a leash for your dog, or something like that, if you tie one end of it to like a wall and you take the other end and you just spin it and kind of twist it up, what's going to happen is that long strand is not only is it going to like twist in that direction, but also the strand is going to start to bunch up in weird ways. We call those supercoils. And so whenever we're unzipping our DNA, we need to prevent that supercoiling or that knotting up of the DNA. And so topoisomerase handles that. The way topoisomerase works is it actually temporarily cuts one of the strands of DNA so that it can kind of like whip around and then it gets put back together. And so topoisomerase actually cuts your DNA, but it only cuts a single strand so that when it unzips, the whole thing can kind of come apart properly.
Yeah, that supercoiling that you're talking about, also rubber bands. I don't know if when you've taken a rubber band off of something and you go to open it, it's just kind of twisted around itself.
Knotted up.
Especially with the bigger or the smaller rubber bands, I've noticed the thinner ones. I've noticed that it tends to happen more with. Drives me crazy, but I don't actually use rubber bands that much anymore, except to tie up my cords when you just have those really long, chargers. There we go.
USB cords and things like that. Yeah.
Yeah. So thinking about this initiation phase, you talked about how we open up, or our bodies, our cells and proteins open up the DNA to start the process. But a part of this initiation phase is thinking about how the protein that is actually going to do the transcription, which we recognize as RNA polymerase, so it is going to create RNA, which is why it's called RNA polymerase. It has to bind to something, or rather that we need something to start the process. And so we call these transcription factors, which are going to bind to what is known as the promoter region. And that promoter region promotes the actual transcription process and is important, both the transcription factors and the actual, which we'll talk about in another episode. But the promoter region is actually really important to get the process started. And so thinking about where this is going to bind, we actually know that we have, rather the promoter region, we know what some of the sequences are. And there's a really famous sequence that you need to know for the MCAT, and that's the TATA box. And it has specific proteins for the TATA box called the TATA binding protein. And so that's TATA. And it's something that you could very well get a question on test day. So it's important to know this specifically. There are other sequences that are important, which we'll talk about in the control. So if you hear us, or rather, if you're thinking, oh, I know some other regions that are important, just hold on. Those will be coming up in another episode.
Yeah. Now, I know you like, just to make clear, in case anybody just thought TATA was a weird name, that TATA is talking about the actual sequence in the DNA. So like thymines and adenine in there. And so that sequence is kind of a place where an RNA polymerase could come in and start this process of making a copy and transcribing the gene. Those transcription factors that we were talking about as well, like, once again, I want to reiterate this because this actually comes up in a lot of AAMC questions. Every single cell in your body has the same DNA, unless you've had like mutations. But like by default, you should assume that the cells in my liver and spleen and the skin on my nose and the thing making my toenail, like they're all those have the same DNA, but they're all making different proteins. And it's because they're getting different kind of orders coming in of like, what proteins do we need? And so that's what transcription factors are. They're things that tell us like, hey, we need more of this protein. We need more of that protein. And so like our RNA polymerase says, okay, let's make a copy of this so that we can get an order to the factories, the ribosomes, to actually build these things. But yeah, so we have a lot of those promoter regions, TATAs being one of them.
Yeah, we didn't mention this, but there are different types of RNA polymerases that you should be aware of on test day. And so thinking about where they're located and what they actually create or promote the creation of, there's RNA polymerase 1, 2, and 3. And you'll see them in Roman numerals, not regular numbers. RNA polymerase 1 is found in the nucleolus, which is an area that's often forgotten about. And so this is going to be involved in the production of ribosomal RNA, so rRNA. RNA polymerase 2 is the one that we think about when we're thinking about just general proteins that are made in the body. And then there's RNA polymerase 3, which is more about other RNAs that our bodies produce. But really the one that you're going to see for general protein production is going to be RNA polymerase 2.
Yeah. And that's a good thing to kind of keep in mind. Is I feel like we often learn about like, oh, there's this protein. And then it turns out like, oh, there's actually a lot of different types of that protein. Yeah. For example, topoisomerase, which I talked about just a moment ago. There are multiple types of topoisomerase. But for the most part, you don't really need to know the differences between those. But just keep that in mind. If you see something in a passage where it says topoisomerase 3A, and it's like, okay, this is just a certain type of topoisomerase. Yeah.
Super quick. That should serve as a reminder that there are going to be things on test day that you will see in passages that you don't know and aren't meant to know. You should know, for example, like you said, that topoisomerase has different versions and a lot of proteins will have different versions, but be mindful of the fact that you are going to see things that you don't know and that you aren't expected to know because the passages are excerpts. So they are taken from other pieces of work, from scientific articles, in the case of CARS, from general pieces that were written in literature. And a lot of times students can freak out because they're like, I didn't know that I needed to know this. And you don't. And so what the MCAT does is it takes what you do need to have memorized and learned, and then will give you some new information and try and have you piece together what you know, the new information, and think about that new information in sometimes simpler, sometimes more complex versions. But that is something to keep in mind and hopefully will keep you a little bit more grounded and a little bit less stressed when you're going through the different science sections.
Yeah. We should take a moment and talk about the two strands of DNA because, so if we have these two strands, like first off, they run anti-parallel, where one is going 5 to 3, the other one's going to be running 5 to 3 in the other direction, right? And so it's like they're running in opposite directions. And they match up. So A's match to T's and C's match to G's. So for example, on one of the strands, we might have CAT, cat. And then the strand that matches that, so if CAT is in the 5 to 3 direction, then the strand that matches that is going to be GTA in the 3 to 5 direction. So the C matches the G, the T matches the A, and the A matches the T. So it's like cat and then GTA is what will match up. One of those strands, like these strands are going to have different names to them, right? So we're going to have what we call the coding strand. Now that coding strand also has other names, and this is a big pain in the butt, and I apologize to you guys on behalf of science. We should not have named it so many different names. This coding strand is often known also as the sense strand, or the plus strand, or the positive strand. I want to be clear. Those are all names for the same thing. Sense, coding, positive, or plus. The other strand that's not the coding strand is what we call the template strand. And so that template strand is also known as the anti-sense, or minus, or negative strand, or the negative sense strand, sometimes you'll hear it called. And so if that coding strand was CAT, then the template strand would be GTA. So the template strand is called the template strand because that's the thing that we're making the RNA off of. So after we unzip this, RNA polymerase is going to come in. And so if my template strand says GTA, my messenger RNA that I'm building is going to match up with that, which a lot of people would say, like, well, isn't that just going to be CAT again? Kind of, except RNA doesn't have thymines in it. It has uracil instead. And so if the coding strand is CAT, our messenger RNA will be CAU. We'll have a uracil instead of the thymine. And so that's why it's called the coding strand, because the coding strand is actually the same sequence as the messenger RNA. It's the actual code that will show up in the messenger RNA, with the exception that thymines are going to turn into uracils. So you want to be really careful with that, because if, like, the MCAT will often ask for the messenger RNA, or they'll ask for a sequence that matches the messenger RNA. And what they'll give you is a strand of DNA, and they'll have to tell you, or they should tell you, it's the coding strand or the template strand. And if they don't tell you, and I think there's a couple of times that the MCAT has done that, it's probably better to assume it's the template strand, because I've seen that come up a couple of times in AAMC exams. And so if they tell you, like, the DNA strand is this, what's the messenger RNA, they're probably giving you the template strand in that. And so you got to be kind of careful, because it's real easy to get twisted around, because, like, the coding matches the template, which matches the messenger RNA, and then something might match the messenger RNA, like a microRNA or siRNA, which we will talk about later on in the future. But you just want to be really clear on, like, what's the difference between those. And like I said, it doesn't help that the coding and anti-coding, or the coding and template strand, have so many names. I didn't even mention that one, but sometimes the coding strand is called the anti-template, and sometimes the template is called the anti-coding strand. And it's just a pain and a little bit tricky, because each one of those strands has like 5 names. And you need to make sure you know which one you're talking about, or which one, more specifically, the AAMC is talking about.
There are also a couple of things that we both haven't mentioned, but that are really important. And so you actually hinted at this, the direction in which you will be given, generally speaking, one of these strands. And that's, you can have either 3' to 5', which is going to be the direction that the RNA polymerase actually creates the mRNA, the messenger RNA. It's going to...
You have to be careful with the direction it builds or the direction it reads.
Other way around. So it's going to read 3' to 5'. And if you think about that, that's because you need to ultimately create the coding, the RNA version of the coding strand. And if it's coding, it's going to read 5' to 3', meaning after the mRNA has actually been produced. And so the RNA polymerase is going to produce 3', it's going to read 3' to 5', produce 5' to 3'. And so I would write this out. This was something that is really easy to get mixed up. And that can actually also give you a hint as to what sequence you've been given. Because if you're given 5' to 3', you're probably looking at the messenger RNA or the coding strand, the coding strand. And then if you're given 3' to 5', then you're probably being given the template strand. Something else that's
you're right.
Really important to recognize and know for the MCAT, which is really important, is the actual differences between DNA, RNA. You mentioned how we have uracil in RNA and thymine in DNA, but you should also be able to recognize the actual structure of C, G, T, A, and uracil, and be able to know what those differences are. It's going to be important. You can have a lot of different questions about the DNA, RNA, transcription, translation, et cetera, which is why we're talking about the very basics here. And so something to be on the lookout for and make sure that you're aware of. Not quite as important as the amino acids themselves, but still pretty important.
Close. Yeah. I want to reiterate that you really need to know your nucleotide structures. To be honest, if somebody asked me to draw adenine, I couldn't do it. So you don't need to be able to draw this from memory, but you do need to be able to look at something and figure out, is that adenine, guanine, cytosine, or thymine? And some of you guys are saying, like, isn't that the same as being able to draw it? Like, not really, right? Like, learn to focus on the differences, right? So adenine and guanine, those are purines, and so both of those have 2 rings in the nucleotide, versus cytosine, uracil, thymine, they all have 1 ring in them. I always like the mnemonic cut the pie, but C, U, and T are pyrimidines, and pies have a single ring in them. And so adenine, guanine must be the purines then. But even then, beyond that, if like guanine and cytosine bind each other 3 times through hydrogen bonds. A's and T's bind twice. And so just looking at a purine, right? Like I can tell it's got 2 rings in it, but you can also just look at this and see like, okay, can I draw a line where 3 different things can hydrogen bond? And if the answer is yes, that's guanine, because guanine hydrogen bonds 3 times. If you can't draw a line where it goes through 3 things that can hydrogen bond and it can only go through 2, then that's going to be adenine. And so I can just like take a glance at something and recognize, hey, that's adenine or that's guanine. And that's really what you need to be able to kind of focus on. And that's what you need to be able to do. But also, you really need to just have a good grasp on those structures, because the AAMC in their practice exams a couple of times throw adenine in there that is connected to other molecules. So adenine has been incorporated into a molecule. And so you need to be able to kind of glance at something and say like, hey, wait a minute, that's ATP. That's in a part of this molecule. And so you do need to have that down as well.
Yeah. So stepping back, we've gotten into some nitty gritty that you need to be aware of, but we talked about how there are different stages of transcription, and that last stage is ultimately going to be termination. And there are some terms that you need to know. So terminators is one of them. The actual termination process can be a bit complex, and so we'll keep a bird's eye view of this. But once we've actually terminated the process of transcribing, there we go, the mRNA and producing the messenger RNA, there are going to be some other things that can be done within the actual nucleus. And I don't think we mentioned this. This is going to be happening in the nucleus. Translation is going to happen elsewhere. But there are a couple of modifications that can happen. And this is where it can get tricky, because I know that in my bio and classes, I learned these as post-transcriptional modifications, which implies that all of transcription has happened. But for the purposes of the AAMC and the MCAT, we don't consider transcription complete until these modifications have been made.
Yeah.
So,
yeah, it is tricky. And this also can create a problem when people are talking about like messenger RNA. Like, if we haven't done these modifications, is it messenger RNA or not? Right. So like talking about it before and after can be a little bit tricky.
Yeah. I've also seen it as pre-mRNA when talking about the piece that has been created before the actual modifications. So in terms of the modifications, there are really 3 that you need to be aware of that help with protecting what will then become the official messenger RNA. And some are going to be thinking about the future proteins that are going to be made. And it's still part of that creation of the paper instructions, like you mentioned. So the first that you should be aware of is the 5' GTP cap. And so this is going to be at the 5' end, and it's going to help protect the actual mRNA from being destroyed. And at the end of the mRNA, you're going to have the poly A tail. And so it's going to be a ton of A's at the end of the sequence. And once you have this to protect the mRNA once it's leaving the nucleus, you're also going to have the process of splicing. And I think this is one that students tend to feel somewhat comfortable with. And that's removing what are called the introns, which are not coding for proteins. And you'll be left with exons, which do code for proteins. And I like to think of this as exons exit. These are the ones that actually make it out, so to speak. And you can also have different versions of proteins, like we were talking about, that have had some exons removed and not others. And so this is how our body can create different versions of the same protein. And thinking about what might be needed, you might have different situations where one version of a protein is produced versus another version of that same protein being produced.
Yeah.
And that's called alternative splicing.
Right. And we'll talk about that a little bit more when we talk about like control of what proteins that your body is making. But yeah, so like after you have created this RNA strand, like you said, sometimes they'll call it pre-RNA messenger RNA. I've also seen it called human nuclear RNA or hnRNA.
Yeah.
And the idea is like it stays in the nucleus and it can't leave until you've done these modifications. And so obviously we're talking about it happening in a human. If we're saying human nuclear RNA, I guess you could have like porcine or pig nuclear RNA if you're talking about it happening in a pig. But yeah, free messenger RNA is a more common name for it, but I have seen hnRNA before as well. And so you put on this, on the 5' end, you add guanine, but you add it not in the normal way, like you would normally add things. It's actually like flipped around and it's added like kind of upside down. And so it's not added on in the normal way that you would add like another nucleotide to this. And then you add a bunch of adenines on the end. And so you add a G to one side and a bunch of A's to the other. And that's the 5' cap and the poly A tail, which is on the 3' end. Those, like you said, they kind of protect the RNA. I always like to think about those things as like a hall pass. It's like a way for the cell to say, like, hey, this is supposed to be here. This is an RNA from the nucleus. And so, like, the RNA, when it leaves the nucleus eventually and goes out into the cytoplasm to go to the rough endoplasmic reticulum, we'll talk about that a little bit later, having a 5' cap and a poly A tail acts as a hall pass saying, like, hey, I'm supposed to be here. Please do not destroy me. And also, please take me to a ribosome. So it is a little bit of a localization sequence to tell me to take me to this thing. And so localization sequences, that's also something that's come up a fair amount on AAMC exams. Note that that's just a sequence that says, take me to this thing. So transcription factors, which we've talked about, like need to go and bind to DNA, they will have a nuclear localization sequence because they need to go to the nucleus. The things that go to the endoplasmic reticulum, like messenger RNA that leaves the nucleus, they don't need a nuclear localization sequence because they're not going to the nucleus. They're going somewhere else. So they need a localization sequence to say, take me to this thing. I do want to mention, like, your cell will chew up random RNA that's in the cytoplasm. And this is actually an important immune thing that we have because viruses can infect your cells, and viruses will often have RNA and DNA in them that they will inject into your cytoplasm. And so your body, if they see random RNA that doesn't seem to have like the hall pass, the 5' cap and the poly A tail, your body's like, ooh, we should maybe destroy this and get rid of this thing. And so that's actually an immune response, is an ability for your body to destroy RNA in your body that might be foreign. So that 5' cap and poly A tail is important. The introns and exons, this, like, you know, if we're going back to like my analogy of the library with the book that says how to build a chair, right? Like what's going to happen is it's going to be like chapter 1, you take the wood and you cut it to these dimensions. Chapter 2, and it's like all of a sudden talking about Goldilocks and the 3 bears, and it has nothing to do with the chair. And then chapter 3 is like, take that wood and like, you know, cut it to these dimensions. And then chapter 4 is something about like the book Moby Dick. And it's like, okay, well this is not part of the thing of making a chair. And so in our DNA, we have these sequences of introns and exons. The exons are the actual instructions. The introns are the stuff that's just kind of got somehow inserted in the middle of these. So the introns are not important and shouldn't be like sent to the factory. Like those things, we need to cut those out when we're making this copy. So we make a copy of this, take all the exons, put them together, and then send that to the ribosome. So the exons exit and the introns stay in the cell. I will say for the longest time, we thought introns were trash and garbage, and we're like, why do these exist? And like, it seems useless. Those introns turn out to be really important for a whole host of different things. Some of them we're going to talk about later. But the spliceosome, the thing that actually cuts your messenger RNA and puts the exons together and cuts out the introns, that spliceosome, the thing that does the splicing, that actually is made of, yes, proteins, but also introns. Like introns will get incorporated into that. And so it's kind of a weird thing that the spliceosome is part RNA from those introns and part protein. It's what we call a riboprotein because its ribonucleic acid is RNA. And so I do want to point out that those exist. I also want to point out that if you're going to cause a mutation in a cell, like if you're going to like mess up the book in the library, if you mess up the introns, generally speaking, that's less of a problem than messing up the exons. The exons are the actual code for how to build this protein. Now, I'm not saying mutations to introns are never a problem, because like I said, sometimes those introns do have important roles. And so mutations to introns can cause disease, but they don't usually cause disease like they would mutations to exons.
Yeah, there's another place where thinking about where a mutation might be important, especially with different, the signals. And so if you get a stop signal really early on in the messenger RNA, or rather in the actual DNA and the template strand, you can think about how a protein might be a lot smaller, may lose really important areas and binding sites. And so that's something else that you can keep in mind that the AMC can absolutely test you on and the MCAT can absolutely test you on.
Yeah. And there's a whole lot of different mutations. I like to stick with that analogy. If it's like, oh, the instructions for building a chair, cut the wood to these lengths, the end. And like, if you put like a mutation that says like, well, stop here, like that's going to be a problem. But also you can have mutations in things outside of even the gene, right? That will change whether or not you're making that protein. Like if you mutate the promoter, which is generally upstream, which is more 5' of the gene itself. And so that gene, like where the introns and exons are, is the promoter is not a part of that. The places where the transcription factors bind, like that's actually not the gene itself. And so you can mutate things like the promoter region, or you can mutate the enhancer region, which is where the transcription factors would bind. And those will affect transcription. And so all of a sudden you might mutate this and you stop making this protein because the promoter's broken. Or the promoter's like way more active than normal, and all of a sudden we're making a bunch of this protein. And that can also be a problem, and that can also lead to disease. We should mention that, I mean, obviously, we kind of talking about disease side of things. There are a lot of diseases that happen because people are making too much of a certain protein or not enough of a certain protein. And so being able to adjust that is important. Also, our body needs to be able to adjust flat out anyway, right? If I haven't eaten in a couple of days, I need to make a lot more of proteins that burn fat in my body. It's like, so I can kind of do that. And so we are going to change the rate of transcription. So we have more messenger RNA or less. Now, we can also affect translation, but we'll talk about that a little bit more in the future. But it's important to understand that these things, there are certain people that get sick because of a certain protein that they don't normally need all the time. And so, like, there are people who have metabolic disorders where, like, if they skip eating certain food, like skip eating, and then all of a sudden they're going to get sick. Or if they eat certain foods, they're going to get sick because it's going to like mess with things. And so there are a lot of diseases focused on this, and understanding this not only helps us understand what makes our body work better, but also pathogens. So we're going to talk about prokaryotes later. Their process is a little bit different, but if we can figure out how to mess with bacteria and not mess with humans, then you're going to be able to kill bacteria and like fight off an infection better.
Yeah. Perhaps we can pull up the image for transcription as we're wrapping up and kind of have that visual orientation and do a quick recap of the different steps in transcription, and just thinking about initiation versus elongation and the termination stages, and then also the post-transcriptional modifications, which actually are part of transcription in general.
Yeah. So normally you'd get like some factor, some signal it could be externally from another source, or it could be internally. The cell is signaling like, hey, I need more of this thing. And so that will affect the transcription factor, which will then go to the nucleus. And once it gets to the nucleus, it'll bind DNA at an enhancer region, and that will cause the promoter to bind RNA polymerase. Now, this figure does not have helicase and topoisomerase in there, but they're going to need to unwind and prevent supercoiling, respectively. And where our transcription or our RNA polymerase is going to go through and make a copy off of the template strand of the DNA. That messenger RNA is then going to go to a ribosome where it's going to be translated, and then we'll have the different proteins there. But it is worth noting that you can have things that bind the transcription factor so that the transcription factor doesn't bind the DNA. And so then we're not going to actually make that protein. But just kind of overall, this is a good kind of like understanding. And I think keeping in mind like where this is all happening, remember that transcription is all happening in the nucleus, right? It's that library of all the different proteins that your body is able to make. And it wants to keep those books there. And it's like, you cannot take these out of here. So if you want instructions for making a protein, you got to make a copy off of our DNA. You got to make a copy off of these books because these are critically important. And we cannot have anything happen to these.
Yeah, you'll see if anyone's watching and actually seeing what we have up. This page is on the Jack Westin page. We have an AMC outline, and we have content that you need to know. And so if we actually scroll, this is the mechanism of transcription. If we scroll down, you can actually see the different steps. And so again, this one doesn't go into as much detail, rather the images don't, but you'll see on the right-hand side that we have little blurbs about the different stages. And so we have the initiation, thinking about the action. It also mentions the TATA box and the TATA binding proteins. And then in the elongation phase, remembering that you are reading, or rather RNA polymerase is reading through 3' to 5' so that it can produce a 5' to 3'. I always have to picture this in my head to actually be able to say it out loud. But that's going to be producing essentially the coding strand with uracil instead of thymine. And then thinking about termination, there are different sequences, which like we mentioned are the terminators, and then those called post-transcriptional, but are part of transcription, which is why they're in here only in eukaryotes, that poly-A tail, the 5' cap, and then thinking about removing the introns, which stay in the nucleus, and keeping the exons, which exit the nucleus, and how alternative splicing can create different versions, of the same protein.
Yeah. As we go on in the future, we're going to talk more about how we can regulate, the rate at which we're building proteins and adjust. So we'll talk a little bit more about some of these things and about alternative splicing, which is, turns out that there's more than one way to splice a gene. But we'll be talking about that a lot more in the future. But hopefully you have a really good understanding of just transcription overall. I do recommend, if you have some questions, you can always go to the Jack Westin page. We have just this is essentially a free textbook of what is the stuff you need to know for the AAMC on these topics. And so note that we do have that there. But hopefully that gives you a better understanding. Next time we talk, we're going to be talking about translation, which is how do we turn that copy of the blueprint into a chair? Right? How do we translate this sequence of RNA into an actual protein that our body needs?
And very quickly, on the page, you'll see that it does mention RNA polymerase has some activity that is analogous to helicase and topoisomerase. But like we mentioned, for some of these, for the transcription of genes, you'll also see that some of them, especially what I've noticed is when papers are referencing cancer, some of these, the topoisomerase is also involved in transcription, helicase, but you'll see that some textbooks mention one, some textbooks mention the other, and this is an area that we're still learning a lot more about. And we just wanted to mention that so that no one gets confused.
Yeah. All right. Well, make sure to come back next time to find out how we make that chair. And we'll go from there. Thanks for joining in. Hopefully that clarifies any of the questions you have on transcription. And if you do have questions, please reach out, podcast@jackwestin.com. We are happy to hear from all of our students.